Good morning. It's June 26th, and this is your daily brief in science. Here's everything you need to know. The James Webb Space Telescope has achieved a remarkable milestone by imaging its first new exoplanet, TWA-7b. This planet is located approximately 111 light-years from Earth and is comparable in mass to Saturn. TWA-7b is notably 10 times smaller than any previously observed exoplanet, making its direct observation a significant breakthrough. Smaller planets are often obscured by the light of their host stars, which adds to the importance of this discovery. Analysis reveals that TWA-7b has a mass about 0.3 times that of Jupiter, equivalent to roughly 100 Earth masses, and a temperature around 320 Kelvin, or 47 degrees Celsius. This groundbreaking discovery was made by a collaborative team from France's National Centre for Scientific Research, Observatoire de Paris (PSL), and Université Grenoble-Alpes. Dr. Anne-Marie Lagrange, an astrophysicist involved in the discovery, emphasized the focus on young stars and the strategy of searching for gaps in debris disks where exoplanets may be concealed. The JWST's ability to directly image low-mass planets was enhanced by its coronagraph, which blocks out the light from central stars, allowing astronomers to detect faint infrared emissions from orbiting exoplanets. This finding marks the first confirmation of a planet in a debris disk using JWST's mid-infrared instrument. The presence of TWA-7b helps explain the gaps in the protoplanetary disk around its host star, indicating interactions between the planet and the disk material during early planetary formation. Researchers are now aiming to locate even lighter exoplanets, potentially leading to discoveries of rocky planets similar to Earth and Mars, which are prime targets for habitability studies. The results of this discovery were published in the journal Nature, highlighting the significance of imaging such a light planet. This discovery not only enhances our understanding of planetary systems, but also paves the way for identifying other similar exoplanets using the JWST. On June 25, 2025, Google DeepMind unveiled AlphaGenome, a groundbreaking AI tool designed to predict the effects of DNA modifications on molecular processes. Following the success of AlphaFold, which won a Nobel Prize in 2024 for its protein shape predictions, AlphaGenome aims to deepen biologists' understanding of genetic variations and their health implications. AlphaGenome has significant potential across various fields, including disease understanding, synthetic biology, and genomic research. Its capabilities could pave the way for advancements in personalized medicine, agriculture, and biotechnology. The model specifically focuses on the 98% of non-coding regions that regulate gene activity and often harbor disease-related variants, providing insights into diseases like cancer. The AI predicts numerous molecular properties such as gene start and end locations, RNA production levels, and DNA accessibility. It utilizes data from major public consortia, including ENCODE and GTX, and can process inputs of up to 1 million DNA letters. This allows for the prediction of the effects of single-letter mutations on gene activity, thereby accelerating hypothesis testing and research outcomes. AlphaGenome employs advanced techniques, integrating convolutional layers and transformers to analyze DNA, achieving state-of-the-art performance in genomic prediction benchmarks. However, it does have limitations, including challenges in accurately capturing the influence of distant regulatory elements, and is currently designed solely for research purposes, not clinical use. Experts like Professor Mark Mansour and Dr. Caleb Leroux have commended AlphaGenome for its ability to address the complexities of non-coding variants and its comprehensive genomic analysis capabilities. The scientific community is encouraged to leverage AlphaGenome in their research, with ongoing improvements and feedback from users prioritized for future developments. The tool is expected to be freely accessible for non-commercial users, with plans for commercial availability to biotech companies down the line. DeepMind has demonstrated AlphaGenome's effectiveness by applying it to mutations found in leukemia patients, successfully predicting the activation of a gene associated with this type of cancer. Recent research has unveiled a concerning trend in the eastern Baltic Sea. the average size of cod has dramatically decreased over the past few decades. Specifically, the average length of these fish has dropped from 40 centimeters in 1996 to just 20 centimeters by 2019. This decline raises significant questions about the health and sustainability of cod populations in this region. Thorsten Reusch, a marine ecologist, highlights that the selective removal of larger fish has inadvertently favored smaller, faster-maturing individuals. This shift has led to substantial evolutionary changes within the cod population. The study reveals that while fishing practices aim to protect smaller fish, they disrupt the genetic and size structure of the population, ultimately favoring survival traits that lead to smaller sizes. The implications of this research are profound. It emphasizes the need for fisheries management to consider long-term generational impacts on fish populations and their genetic diversity. The ecological consequences are troubling. Smaller cod mature earlier but produce fewer offspring, raising concerns about the overall decline in population numbers. Despite a complete fishing ban enacted in 2019, current data shows no signs of recovery in the size distribution or overall population of eastern Baltic cod. This population of cod has a unique lineage, having diverged from others 7,000 to 8,000 years ago. Findings published in the journal Science Advances suggest that both genetic changes and environmental factors are contributing to the lack of recovery in cod size since the ban. Marine biologist Prof. Stefano Mariani emphasizes the importance of genetic analysis in understanding these changes, while also noting the role of environmental factors. The evolutionary trend favoring smaller cod could complicate future recovery efforts, hindering restoration initiatives. Researchers utilized otoliths and high-resolution DNA sequencing to analyze age and growth patterns, revealing that human activities have induced rapid evolutionary changes leading to reduced size. Furthermore, the decline of cod impacts its role as a top predator in the Baltic Sea food web, limiting its ecological function, especially as other fish species like herring and sprat also face overfishing. To effectively protect the cod population, a significant reduction in fishing pressure is essential, adhering to sustainable usage principles, though practical implementation has been lacking. A recent study from Japan reveals promising insights into the role of vitamin C in skin regeneration, suggesting it may have the potential to reverse some aging effects on the epidermis. Researchers focused on human epidermal equivalents, which are advanced 3D culture models designed to closely replicate real human skin. This allowed for a detailed investigation into how vitamin C affects epidermal structure. In the study, skin cells were treated with vitamin C at concentrations typically found in the bloodstream, providing a realistic context for assessing its impact. The results showed that vitamin C treatment led to a thicker epidermal layer and increased keratinocyte proliferation, while the outer layer of dead cells remained largely unaffected. Notably, the research also highlighted a significant rise in global levels of 5-hydroxymethylcytosine, a marker associated with active DNA demethylation, with levels increasing 5 to 7-fold in treated samples compared to untreated ones. Additionally, over 10,000 DNA regions displayed reduced methylation, with 12 key proliferation genes showing substantial increases in expression, ranging from 1.6 to 75-fold following vitamin C treatment. Among the identified genes reactivated by vitamin C was ROS1, emphasizing the vitamin's essential role in cell growth and tissue maintenance. This study was made possible through grants from the Japan Society for the Promotion of Science, specifically under grant number 19K05902. A recent study published in Nature has unveiled a striking mechanism by which cancer cells bolster their survival and spread, by stealing mitochondria from nearby neurons. Researchers from the University of Texas MD Anderson Cancer Center, including Anand K. Singh and Yuen Pan, conducted both in vitro and in vivo experiments that revealed breast cancer cells acquiring mitochondria from neighboring neurons. This acquisition leads to an increase in mitochondrial mass and enhances energy metabolism within the cancer cells. The study also emphasizes the significant role of nerve infiltration in tumors, linking it to aggressive cancer behavior. Denervated cancer cells, those lacking nerve support, exhibited reduced growth and altered metabolic profiles. In experiments involving mice with aggressive breast cancer, blocking nerve function resulted in diminished metabolic activity of the cancer cells, highlighting the influence of neurons on tumor metabolism. Elizabeth Rapaski from the Roswell Park Comprehensive Cancer Center characterized this discovery as a major advancement in cancer neuroscience. The findings suggest that inhibiting mitochondrial transfer could potentially hinder the spread of lethal tumors, prompting researchers to consider drug development strategies targeting this process. Additionally, the study underscores the role of tumor-associated neurons in facilitating cancer cell migration and metastasis, particularly in breast cancer. Co-author Simon Grellett from the University of South Alabama noted that targeting this mitochondrial theft could open new avenues for treating metastasis, a key factor in cancer lethality. Clinical samples from prostate cancer patients further supported these findings, showing a correlation between perineural invasion and increased mitochondrial load in cancer cells. Notably, 14% of tumor cells that spread to the brain contained nerve-derived mitochondria, indicating these cells may possess enhanced capabilities for metastasis compared to those without such mitochondria. This has been your Daily Brief in Science. To read more about these stories, follow the links in the episode bio. You can also subscribe to these updates via email at www.brief.news. For more daily podcasts about the topics you love, visit brief.news/podcasts. Tune in tomorrow. We'll be back with everything you need to know.